Yeah.
Any other announcements? All right, I'm going to turn it over to our retired professor here to introduce the speaker.
What he's doing. Thanks, Michael. It's a real pleasure to have our seminar speaker here today, Melissa Hawkins from Humboldt State University. She brought with her one of her graduate students, Stella Ewan, who's there doing some work in the collection while they're here. Missy just arrived in Humboldt a year ago in the fall, right? 6 or 7 months or so pregnant, and so I think her first semester was maternity leave. Yep. So now she has a 16-month-old. She's assistant professor in, what is it, biology department?
Yeah, it is.
In the biology department, and curator in the Vertebrate Museum, which is a great institution for a relatively small university setting. I know that institution well because I visited there a lot over the years. Missy got her degree from George Mason University in 2015. She did most of her thesis work, I think, at the Smithsonian and probably the National Zoo Lab with Jesus Maldonado and Chris Helgen in the mammal collection and other people there. She's had an amazing career so far in the field, working in Malaysian Borneo for much of her thesis. And then she did a postdoc work with Ed Lewis at the Dooley, Omaha Museum, Zoo, where she spent several long field seasons in Madagascar working on everything from eyes to brown lemurs to sifacas. She's a genomics whiz, using all the kind of modern molecular tools and analytical tools that are now available. And that's, I think, probably what she's going to summarize today over these various kinds of critters that she's worked on, from cool rats in the montane areas of northern Borneo to the Sulawesi radiation of squirrels, which on a small island has 3 genera and 20-some-odd species that are all endemic to that island, as well as the Malagasy lemur biology. So Missy, welcome to the museum. I hope this is the first of a long series of visits over the years that you're in Humboldt, and we look forward to hearing.
Great. Thank you, Jim. It's so exciting to be here. Me and Stella are going to have to be kicked out, I think, at the end of the day today, because it's really nice to spend blocks of time in the museum talking to all of you and hearing about your research. So much active field work going on, which of course keeps my heartstrings again, wanting to get back in the field. You guys all know this because you're field biologists, but you forget about all like the terrestrial leeches and ticks and mosquitoes when you're back for too long. And I've clearly been back for too long, so I'm very aching to get back in the field. So I'm going to talk about a couple different case studies that I have selected to show some of the breadth of the research that I do. And I have put little icons so you can follow along if you're dazing off of how boring the methods are, so you can keep track of which project I'm talking about. So Jim summarized this probably better than I could. I consider myself a mammologist, although I have a master's degree on snake genetics from Central Illinois. I worked on prairie ringneck snakes for my master's thesis, but I always knew I wanted to get into mammals. But I still have worked on a lot of projects, including both birds and reptiles. But the methods are all the same. DNA is extracted in a similar fashion, and you can do all the same fun sorts of things with evolutionary biology and biogeography. This is a fun little cartoon I always put up about a bunch of penguins complaining about the cold, talking about his calculations that Antarctica will be at the equator in 50 million years, and the one surly penguin is saying that we should have just followed the lemurs and joined them on Madagascar and avoided Antarctica. So I like to show that to a lot of my students. I like to work in biodiversity hotspots because it's just more interesting when you have complex systems. And unfortunately, I haven't been able to go to Sulawesi, so the research I'm presenting is completely collections-based research for the Sulawesi chapter. But as Jim mentioned, my dissertation was based on, the full project of my dissertation is still not published. I know a lot of people can empathize with that in this room. That it was supposed to be the phylogeny of the subfamily of Southeast Asian tree squirrels. There's 60 plus named species, but many of those are species complexes. And I've published several chunks of that, but not the whole culminating paper yet. So after this postdoc paper, that's what I'm going to switch gears on and hopefully have an exciting talk in the future about what we've been recovered on the biogeography of the Southeast Asian tree squirrels. So I have a lot of focal research questions, and I'm going to go through these rather quickly. The first one that I'm really interested in is how do traits and species diversify within an adaptive radiation, and along with that, how does the geology really influence those kinds of speciation mechanisms in these different island systems? And then how does global climate change affect the genetic diversity, but also the ranges and threats to movement patterns of montane species? And the last one that I'm not actually going to talk about today is how does the tempo of evolution affect population size through time? I will highlight a paper that I published that goes through that in a little more detail, but the first three are really what I'm going to talk on and hit with the different case studies today. So I'm in a room of people that are very familiar with this technology, which is lovely, but just to really quickly get everybody up to speed of what I'm talking about in terms of methods, almost all of this work has been done using high throughput sequencing methodology, and like probably many people in here, I'm using almost exclusively Illumina sequencing for reduced representation of the genome, not whole genome sequencing. Now we're switching for whole genome sequencing to do other longer fragment type sequencing. But I used Illumina to do both mitochondrial genomes in collections. It's a lot easier to do mitochondrial work because of copy number in cells, and you can get that relatively easy. And nuclear markers, which we've been using ultra-conserved elements, are a little more tricky and have a lot higher dropout rate just because of copy number and degradation of museum specimens. So ultra-conserved elements might sound like a terrible phylogenetic marker, right? Kind of an ultra-conserved part. But for those of you not familiar with them, this is a little plot showing the amount of variable sites in UCEs. This is averaged across several thousand. And what you see here is this little black line indicates the ultra-conserved region. And the reason they're so nice is that you don't have to develop species-specific markers for use on a broad range of taxa. So Travis Glenn and Brant Faircloth have done a lot of work on finding panels of UCEs for a variety of different taxa. The one I use is for tetrapods, so I could use it on pretty much anything I've ever worked on, which is lovely because it's a lot of time and cost involved if you're designing your own panel for either SNPs or just whole genomic representation. And there's no reference genomes for any squirrels anywhere closely related to the ones I work on. And as you go away from this core site, you can see the amount of variation gets quite high. So you can get really well resolved phylogenies. Some of them have no variation in certain groups, but you can find a lot of loci by throwing 5,000 and a panel at something. You can get a lot of variation. And I've had really good luck resolving phylogenies with UCEs. Going along with that is a lot of bioinformatics. And I'm not really going to talk about any of the bioinformatics here. I've done some work on optimizing methodology for museum specimens. But I think the stories of the critters are more exciting for especially a crowd like this that works on really cool, diverse animals. But I'm happy to chat about bioinformatics if people have questions. But I am by no training a programmer, just by necessity have learned a lot of how to do a lot of these things. So for UCEs, we do in-solution capture. I'm putting things up here so when I switch from jargon to jargon, you'll know I'm talking about the exact same thing. In-solution hybridization / capture is exactly the same thing. It's a lot more efficient than doing PCR, especially when you're talking about 5,000 markers. Because it's not nearly as specific as PCR, you can have your whole DNA extract, do library prep on it, so you're adding the sequencing primers and an individual barcode to that sample. We are doing dual index TrueSeq style adapters so that each side of your fragment has a different 6 or 8 base pair code that you can sort it from later. And then what we do is you take your baits, which are biotinylated here, and hybridize the fragments of interest with them. Because of how hybridization works, they're long probes, so sometimes 80, sometimes 100 base pairs long. You can actually make your own, or you can buy them from commercial places. And because of the length of it, you can have a lot more mismatches and still have successful enrichment of your sample for in this case, the orange pieces of DNA that you're interested in. And then you use magnetic beads, which is one of the most exciting things when you have clear liquid in tubes you pipette all day, and you can actually see something happening when you have magnetic beads sticking to the side of a tube. You at least feel like something's happening. Then you're recovering only the biotinylated molecules that have hybridized to theoretically the genes of interest that you're working on. And you recover them, and then you do limited cycle amplification to make enough copies to adequately sequence. But the really simple version is like you're picking the red apples out of a bunch of red and green apples, if the red apples are the ones you want. You can design these. I've designed a number of arrays. Sometimes I've worked on a project that designed an array to enrich host parasite pathogen from tick samples, so you could get the mammalian host or avian host, any type of disease that was in the tick's salivary glands or gut, and the host or multiple hosts that they've had. So you can do a lot of really cool creative things with this for single species or for large amounts of species. So it's really changed the way we go about lab work. So again, here's the 3 case studies I'm going to talk through. The first one is the colonization patterns of the Sulawesi squirrels. The second one is from my postdoc looking at the phylogeny of the sifakas. And then the final one is a project we're just getting ramped up to work on now in Northern California, looking at the evolutionary history and movement and speciation processes in the redwood chipmunks in Northern California. So the Sulawesi squirrels, I am far from the expert of Sulawesi in this room. There are many people here that have been to Sulawesi and I have not. Been to many places over decades. But this is again done from solely museum specimens. And one of these days I will get to Sulawesi. I have to get to Sulawesi because it just seems too incredible. This was supposed to be included in that big phylogeny of the subfamily of squirrels, but I got too interested in Sulawesi and I said, come on, let's just do this one as a separate paper. I got that out in 2016 in BMC, Evolutionary Biology. So Sulawesi, again, as many people in this room know, is a really cool, complex cage-shaped island in Indonesia that is southeast of Borneo and between that and Papua New Guinea, and it's kind of this melting pot of arrival and dispersal from different areas, some from Australia origin and some from Southeast Asian origin. And the interesting thing is, while sea level change has been really important in the Sunda Shelf, allowing for connection and disconnection of the land masses, there's a deep trench here that has never connected Borneo to Sulawesi, for example. So it's only ever been colonized by over water dispersal. And as Jim mentioned, there are 3 genera of squirrels that are described on Sulawesi. This is a rough approximation of their ranges. We have 2 species that are widespread across the whole island, this pink color here and the dark green. And then others that have much more restricted ranges. There are some with disjunct ranges like this Hyoscyrus species. I don't buy that that's going to be the same species. And then within the genus Procyrillus, which is these two maps, there are a variety of habitats that are disjunct and probably have a lot more diversity than we currently recognize. These three are not included in the genetic analysis in this study, the light colors and then the one from the islands off the north coast. So of course, like everything, there's a lot more work to be done. I actually went online because I always say, oh, you can't even Google good pictures of these squirrels. And I tried again last night, and I found a decent one of Procyrillus murinus from like 2015 or 2016. And then this one, I believe, is Procyrillus leucomus, neither of which are drawn in this illustration from Guy Musser's monograph that really looked at the morphology and identified a bunch of lice that were endemic to these rats. He's retired from AMNH. And so I was pretty happy. I found two squirrels that I can pretty confidently say are from Sulawesi, because sometimes you'll Google Sulawesi squirrels and get very obviously Bornean squirrels and such. So they're an adaptive radiation because this squirrel is very small. It's a little pygmy squirrel. So yeah, you can do the oohs and ahs because they're really cute, tiny pygmy squirrels. Whereas the rest of the genus Procyrillus has a lot of this ornamentation. This species has two named subspecies, but it has these white patches behind its ears and black ear tufts. And the belly's a little bit bright orange. This one, the illustration here, kind of looks similar to that, but it has white ear tufts, and that's a different species. So for squirrels, they actually have pretty good color variation in their pelage for that genus. This is an illustration of Rubricyrus, which is the giant ground squirrel, and they are bright red, so this picture doesn't really do them justice, and that one I can never find good pictures of, but they're quite large. And then the other genus, which also has no pictures, is called Hyoscyrus, and it has this extremely elongated snout, and they are earthworm specialists. It has a really long, thin snout, and Hyo means pig in Latin, so it's this long-nosed squirrel, and that contains two named genera. Rubricyrus contains one, and everybody else is put into Procyrillus. So we knew from the squirrel-wide phylogeny that they were colonized probably a single time, but what I wanted to do was to look at a lot more markers. The published study was 16S mitochondrial and 12S mitochondrial, I believe, only. So I wanted to add a lot of nuclear loci to it and see if we could kind of figure out the movement patterns across the island through time. So for the mitochondrial genome, we have more taxa than you'll see in the UCEs because we have a lot higher dropout, as I mentioned, with UCEs. And interestingly, here we have our Bayesian support from this beast tree, so a dated tree using our outgroups from other Southeast Asian squirrels. This Lariscus actually came from the MVZ's tissue collection from Sumatra. That's kind of fun. And then the dates here are in millions of years. So you can see these are pretty, for mammals, these are pretty old radiation. And we can see all of the Sulawesi squirrels are the colorful ones. And here our dated split in the mitochondrial genome is 9.7 million years ago. And interestingly, you see this paraphyly between Procyrillus murinus, that little pygmy squirrel, and the rest of the Procyrillus, which are all up here, with the two other genera of Sulawesi squirrels in between them. So of course I got the mitogenome results first, and I was like, oh my gosh, do I get to name a genus of squirrels? And then I did the nuclear data, and then they popped back in where they should be with the rest of Procyrillus. So no, I didn't get to name it a genus. It's not that it's not interesting to find this deep mitochondrial capture event, but the nuclear data show that they are one monophyletic group for all Procyrillus and for all Sulawesi squirrels. So based on this, this is from minimally 1,000 UCE markers, Unfortunately, my other species of Hyoscyrus that I sequenced only got 258 UCEs enriched. So I took that out of the analysis because in the mitogenome they came back as sister species, but here you have a 5.7 million year divergence from the mitochondrial genomes of those two within the same genus. And you can see that there is quite a deep history within species for both mitochondrial DNA and the nuclear DNA. Within the Procyrillus leucomis, we see 4.6 million years of nuclear time that has passed within them, and this one is distributed in the, I'm all over the place today, is in the northern peninsula. So this is a slightly outdated paper now that was just brought to my attention yesterday, that there's new geological information that came out in 2019 or 2018, remapping some of the movement of Sulawesi and what was above sea level at different time points. But based on this reconstruction, and our dates wouldn't change from that, obviously, because we use fossil calibrations from other Southeast Asian squirrels, that our colonization of Sulawesi looks like it's around 12.5 million years ago. And we assume it came from, it's most likely it came from Borneo just because it's most geographically proximate. And then what's interesting is you see the first genus to break off is the Hyoscyrus, then the Rivercyrus, and then between 7 and 5 million years ago you see this big kind of explosion of within species, within genus diversification for Procyrillus. And I likened that to the mashing and uplifting of the land mass to allow for more different niches to exist and allow for specialization from this genus. So I might have to rethink that a little with the new dates that came in, but it's still pretty deep, so between 7 and 5 million years ago, a lot is going on within that genus. So for that one, we see a pretty ancient crossing of Wallace's line, which is different than a lot of the classic examples of adaptive radiations, meaning things like the African cichlids and Hawaiian honeycreepers. They're very young comparatively to this adaptive radiation. We see the ancient, oops, I put the wrong species here. We put ancient mitochondrial capture in the pygmy squirrel. And we found deep divergence within the species that we had multiple samples of, which is obviously biased by the few number of samples we had, but it leads us to a lot more questions on what was the movement patterns through time, where are the breaks between these subspecies that might in all likelihood be separate species. And I looked in a lot of different methodology to see if there was gene flow between the pygmy squirrel and the other species that share the nuclear signal but have the very different mitochondrial signal. And it seems that there's no ongoing gene flow, so we think that these things had a mitochondrial capture event from either one of these unsampled lineages or some extinct squirrels, and that just has retained through time in their mitochondrial genome but not reflected in their nuclear genome. So now, of course, every time you publish one paper, you want to publish more papers in the same system because you have more questions and answers, right? We love to do whole genome sequencing. I know a lot of you in this room know Kevin Rowe and Jay Gesselstein. I've talked with them before because they're all very interested in the shrews and rats and less interested in the squirrels, which is great for me because we are going to work together. We've been in talks before about doing whole genome sequencing on the tissues they've collected in their trips to Sulawesi. So they have each genus represented, which is great for looking at this adaptive radiation. I'd love to do fine scale sampling and expand from museum specimens and look at where are these areas where we have a lot of samples, and can we look at maybe the movement and ancestral reconstruction of these species and genera through time and space. So that's some of the work I'd like to do on that. It's on my ever-growing to-do list, but everybody in this room knows about that. So then I'm going to talk now about work I did for my postdoc, and it's working on the phylogenomics of the sifakas. I never want to act out what a sifaka looks like when it moves around, so I put in a gift for you guys. This is a Burroughs sifaka. They're the only, the sifakas are the only mammals that use vertical climbing and leaping as a locomotion pattern, and these ones are incredibly skilled, is what you can't really tell in this image, but they can jump from these really prickly cactus from one to another over 10 to 15 meters in a single bound, and somehow they can grasp it without impaling their hands and feet on this cactus. So they're an interesting group that has been studied in a lot of different manners, and we were the ones that wanted to look at the phylogenomics of this genus. Because again, there had been some mitochondrial phylogenies put together. A lot of them lacked some individual species, and some of them lacked resolution. And obviously that makes sense if they're a group that either recently radiated or if you just don't have enough information in your mitochondrial gene. Historically, sifakas were described, the type of the genus is the Diademe sifaka, from one individual that was sent to the British Museum and described in 1832, I believe, or 34. So very early description. The type location is Madagascar. So not particularly helpful for this fine scale work that I'm trying to accomplish here. And then after some time in 1870s, the Burroughs sifaka was described. So they started off being described as two species, and since then they have been split in much further than that. But they had historically documented in the mitochondrial signature that this species, the silky sifaka, had mitonuclear incongruence with one of the species closely related to it. So it has a mitochondrial capture event as well. So this is where they live. Across most of the forested regions of Madagascar, there's not a whole lot of records from central Madagascar, but again, there's not a lot of forest in central Madagascar. It's more rolling plains and grasslands. It's very controversial whether that's because of complete anthropogenic modification of the landscape, or if it's because that's just the way it has been and natural aridization of the island. But down in the south of Madagascar is the spiny forest, where I just showed you the Burroughs sifaka jumping up and down in the trees. In the western area, you have like kind of these tropical deciduous forests, which is really cool. And then the eastern part of Madagascar is really the tropical rainforest areas of Madagascar. So you see different species in these areas, and they're pretty much broken into the western species, which is this group here. This is for the younger people in the audience, younger than me. Zaboomafoo was like a kid's show apparently. When I teach mammalogy, they're like, oh yeah, Zaboomafoo. I'm like, I have no idea what this is. So I had to Google it, and it's like some brothers that have a lemur and they have adventures. I don't know. It's not on the air. It was a PBS show for a while, but it's not on the air anymore. And then we have our eastern group of sifakas, which includes this one up far in the north and these three species. And then this one, the Tattersall sifaka, was actually not described till like 1986, which is really strange because it is pretty distinct both phenotypically, morphologically, and genetically from the others. And what you can see here is obviously they live in this one, there's less than 100 individuals of the entire species left. So they live in really restricted areas. They are historically eaten in certain parts of the range by the Malagasy, and I believe all of them are listed as either endangered or critically endangered. So we wanted to understand more about the phylogenomics of this species by adding a lot of nuclear data to the mitochondrial results that were already published. So we actually I was very fortunate that the group I worked with for my postdoc has been in Madagascar for over 20 years and has exported these CITES 1 listed samples before I got there. So it wasn't contingent on getting CITES permits for my postdoc, which, as a lot of you know, can be a really lengthy process to get out of any country. It took about 9 months for the U.S. Fish and Wildlife to even look over an application, and then you have to have the U.S. one before you can get the Malagasy one, and it's a long process. So they had done an export in 2009, which had a lot of the sifakas that they'd already collected from over 200 sites. They've sampled lemurs in Madagascar. So we used Illumina HiSeq and NextSeq to do two different enrichments, one for UCEs and one for a custom gene set. I haven't finished going through the custom gene set yet, so I'm presenting on the UCEs here. And here's a very pretty zoomed out image of our tree. We didn't have any super surprising results with the eastern versus western split of the species, except we did confirm that this species, the crowned and deccan sifaka, have active hybridization going on. But if you see our distribution map and sampling locations, we have very biased sampling sites for those two species. But it seems like they are, at least in these contact zones, they're actively hybridizing, and they've known this based on physical appearance of some that are hybrids. These are quite different pelage, so you can see and easily identify a hybrid animal. Whether it's only an anthropogenic hybridization because of forest contraction, we don't really know. And that's one area we want to expand our sampling to figure out what's going on in the rest of their range. But what I think is actually the most interesting thing here is that the eastern group of sifakas, which are shown with their little pictures and colors, in the diademe sifaka, we found two different clades within it. So if we zoom into that, we look at the plots here of who belongs in which clade. And I thought for sure it was going to be like, okay, the ones that are most close geographically are going to fall into one versus the other clade. But then it didn't really make much sense, and we thought, okay, well maybe it's high versus low elevation, and then we explored our data and it wasn't really associated with that either. And every way we break up our UCE data, we recover these two clades, so it seems fairly robust. So we did this ecological niche modeling, which I had to learn for this because I do not delve much into ecological niche modeling. But there's been papers published on climate zones in Madagascar, and that's what all these crazy colors are, is different climate zones that have been identified. And then we plotted our samples here, and what you can see is that we have two clades of sifakas corresponding to two ecologically different niches, which was really clean and amazing. I did not expect to actually have any kind of resolution at this level. And then if you look at each clade independently, you can see that there's probably some sort of competition that limits them to those different clades, or the ecological niches. So they correspond really neatly, and some of them you can see here, there's little dots of this light blue that's retracting, and we have a blue dot up here. So with climate change and deforestation, we're probably going to get some artificial populations that are falling in the other current distribution of these different niches. But it was a really surprising find, and I think our most significant find in this study. And it brought me back to that type literature to try to figure out, okay, has there ever been subspecies within Diadema? Because, as I mentioned before, there was only two named species originally, which included all eastern species, and they were all subspecies of Diadema. But within Diadema itself, there's never been another subspecies named. So we're in the process of trying to figure out, when you have a type description that says Madagascar, you can't exactly figure out which of the two clades the type specimen belongs to. So this might turn into a snowballing of trying to get the sample from the British Museum, from the holotype, and see if it corresponds to one of these two clades. But it was a pretty exciting result because no one's really documented this morphologically. Because the Diademe sifaka is known to have quite a bit of pelage variation, but in other species, related species like the Indri, the color variation means nothing in terms of the genetics that's been done before. So we found some cool things. There's a lot more things we need to do with this, and I am not a geneticist that wants to just keep splitting everything for splitting's sake. So I'm trying to figure out, like, if we think these are more ecomorphs or subspecies, But of course, as everyone in this room knows, it's very difficult to make a judgment call on that. We need to look through a lot of the enrichment data for the custom genes. We have found in the low-hanging fruit of mammal pelage is the MC1R gene, and we found what we think are the 2 different mutations responsible for the 2 black species of Sifaka, which is kind of cool. And then now what we're doing is, of course, I'm on my computer right now running Beasts to try, for a week I've been running Beasts, to try to figure out the dates between those different clades, but also for the whole tree as a greater, as the genus. We are running getting Mr. Bayes finished, and then I want to do a Bayesian skyline plot because my original attempts at skyline plots have shown a different demographic history for these 2 different clades. So trying to figure out, like, there might be some different rates of evolutionary trajectories that those 2 are going on. And I currently have 2 undergraduates that are working on the external morphometric data that they collected at the time of sedating every lemur. A critically endangered species we're not going to go out and collect 200 of. So they're all external measurements, so I'm assuming we're going to lose a bunch to mismeasurements, but we're going to see if these 2 clades correspond to anything morphologically that might strengthen our arguments of them being subspecies. And then it would be lovely to get more sampling of the crowned versus deccan Sifaka that do have this active hybrid zone. Okay, so the final project I'm going to talk about is on North American chipmunks. So I'm at Humboldt State. We're in the middle of the redwoods, and there are a lot of chipmunks. There's 25 species described within the greater genus Tamius, which has since been split into 3 subgenera. You don't like it, Jim? You're shaking your head. But in California, there are 13 named species of chipmunk. So there is a lot going on in chipmunks in California. Some of these they all look pretty similar, right? They have black and white stripes on their backs and usually some amount of white on their face. But some of these are from Russia, and some of them are from our backyard here. So there's a lot of convergence on this body plan. So it's quite difficult to tell them apart, which is why they were all 1 genus and now 3 subgenera. So the subgenus for the North American species is Neotamius. I might slip up and just say Tamius because I'm more used to Tamius or Eutamius than Neotamius. But this is the distribution of the Townsend's group of chipmunks, which are considered the redwood chipmunks. And you can see here we have Canada, Washington State, Oregon, and California. And we have a lot going on here, which is predominantly bounded by watersheds and river boundaries. And obviously Northern California has a lot of topography going on. And you can see if you look at the different rivers that they have pretty roughly followed some of the major rivers, but not all of the major rivers, withdrawing the boundaries for these different species. We have a couple scenarios of what would possibly happen for speciation events, right? This is a little goofy of a cartoon, so bear with me here. If we historically think of them as one nice panmictic species, you don't see any differentiation between them. But if you have something like a river barrier introduced through glacial times, you might still not see at really recent splits any sort of differentiation in the phenotypes, but you might through time have some different physical appearances. What seems to be happening, at least in the populations we have looked at, is that within species there is quite a bit of pelage variation, but nothing that really neatly corresponds to one species or the other. So we're not really seeing any phenotypic changes in the external morphology, and I don't think there's been any discrete characters on the cranium that can be used to identify them. The only thing that's used right now is the bacular morphology. Again, sometimes in non-scientist groups, I just gave as part of this talk to some retired faculty in all kinds of departments at Humboldt, and you can see the non-scientist men start squirming a little when you talk about a bone in the shaft of the penis. And for whatever reason, there's a lot of diversity in these phenotypes. A lot of it is thought to be a species lock and key prezygotic isolation mechanism. And you can see some different groups of mammals, like these are some skunks, two different subspecies and subspecies of skunk bacula, look very different. These are, I think, the scariest looking ones. I think they look like a lamprey mouth or something, and those are different types of tree squirrels baculum. And then they're only found in specific types of mammals, rodents, insectivores, and carnivores. And when we look at, these are just a handful of the Townsend's chipmunks I pulled out of the drawer. You can see some that have a little bit of phenotypic variation, but if you pull out more and more drawers of them, you'll find that it's vastly overlapping. But in the baculum, there's a lot of diversity in these animals. And there's been a little bit of work done looking at the call structure of chipmunks, and that they are variable as well. So this one I think looks like a little Adirondack chair. But again, and these are millimeter marks, so these are like 2 millimeters long. So historically, people would do linear morphometrics from like 10 to 12 landmarks on these 2 millimeter long things. So some of them have this one we call the spoon. And then Senex and Ocrogenes look very similar, but you can statistically tell them apart after doing those different sets of morphometrics on them. So I am working with Dr. Karen Rice, who's at College of the Redwoods, and she does the bacular portion of the work because, and she's familiar with micro-dissecting these and staining them and all of that. So I tell her, just give me a piece of tissue. I can't measure something that's 2 millimeters long. So again, we've mentioned that it would be lovely if these worked as a very nice lock and key mechanism where some species just cannot physically breed with another because of the baculum shape. But in chipmunks, particular in North American chipmunks, the more studies that have looked into this, the more studies we've found that totally refute this and say, oh, well, they not only can they hybridize with various different species, but they have fertile young, so they break all of our species concepts. And I think it's something due to a post-glacial expansion in Northern California that perhaps when this phenotype of the shape of the baculum evolved, they were true separate allopatric species, and now they've become sympatric following the retreat of the glacial ice sheets, and it's confounded our understanding of the group. This is a very unresolved mitochondrial tree that shows Tamias senex all over, but part of the problem here is we don't necessarily know if published sequence have named something Tamias senex because of the location it came from, or if they have really gone through and finally identified bivacular morphology, which most of the time isn't the case. So it's pretty much a mess. You can see all of the species are pretty much admixed, and again, this is only mitochondrial DNA. So we are going to use fancy, well, Karen calls it Missy's genomic magic, which I kind of love the ring to that, to see if we can detect ILS and hybridization and what really is going on in these species. So last summer we went out and did our first field trip, had 3 students, myself and Karen and my 6-month-old at the time, and went and got some chipmunks. So this is me training them on how to handle chipmunks in the field. And what we want to do is look at this genomic data and look at the splits between these species at very strategic locations on opposite sides of rivers, because that is what has been used for identification of the range of these animals. So if we look at the map, this is where we went last summer in Shelter Cove area. And so here's the Eel River watershed, and we're sampling on all sorts of these locations opposite sides of the river this summer, bringing Stella with in the field. And we're going to try to get as many chipmunks collected as we can. And we're also putting in for the Cascade Siskiyou National Monument in Oregon. They have a grant that came up, so we're like, okay, let's try to see if we can get some the grant for that and some permits to go get some, which should be Neotamias siskiyou from that location. And I have a new graduate student starting in the fall to start working on this project. And then we'll just have to keep finding the different areas of opposite sides of rivers to try to figure out the bounds of these different species. We're also working with Matthew Dean at UC San Diego, I believe, and he is going to be working on micro CT scanning. We sent 10 bacula of these two species. We figured they're really different. He does work on Peromyscus, and this is a picture from one of his papers that uses computer modeling to correct for shapes. As soon as we sent him a picture of the squirrel bacula, he was like, oh gosh, this is a lot more complicated than the Peromyscus. So he's going to test his software and see if he can tweak his software to be able to analyze the 3D structure of these. So hopefully we can use that and continue collaborating with him and figuring out where the diversity is, if there are weird intermediate forms of the bacular morphology. So that is still to come. And then this is a picture of one of the quintessential me holding an endangered lemur that we were darting in the field, and you get all this, rush of adrenaline when you're with a team that gets to knock out and recollar one of these animals. And obviously I had someone else took this picture of me, and I was really excited because they had just darted a second one. So they go, oh, here, hold the lemur, which always breaks your heart when you're in Madagascar studying lemurs, when the dart team says, hold this lemur for me so we can go get another one. And I was just like having a moment with Griff here, and then he released his bowels all over me, which also was captured, and he's actually sticking his tongue out, which so I think he was maybe more aware of what was going on than we gave him credit for. So this is one of my funny series of how to end a talk, is me getting pooped on by a greater bamboo lemur, which I obviously haven't talked about today, but we published the genome of this species, and I'd be happy to chat about that. I have a bunch of people to thank, a bunch of organizations. Thank you guys for having us here, and I'd be happy if there's time to take any questions.
So the question about the two clades and the one lemur species, why aren't you thinking about calling them different species? Because it looks like their divergence time was kind of on par with the other ones there.
Right.
The only difference is that they didn't have a strong morphological difference that you can detect right now. But I mean, cryptic variation can be as real, and they're coming so close together that, given that the other ones hybridize, I mean, at least in the gene flow, they seem to be good species.
Yeah, we haven't ruled that out, but I've worked with a few primatologists that are super eager to split every single thing. So I refrain from saying new species of lemur before we have really quantified the date. And we're going to see if we can get morphological in a decent time span. Then yeah, I mean, some of those sites are probably 20 kilometers apart. I don't know if they would really be in contact because the forest is probably gone between those two spots. And there's almost no diadem siphacas in captivity, so I don't know if you'd be able to see oh, these ones just won't breed together, maybe they belong to different clades. But yeah, I haven't ruled that out. I just walk a little more carefully, especially in primates, was just gallivanting about a new species. But it'd be interesting, I think, with the skyline plots and the dates to see oh gosh, how long ago did they actually split?
And you can model gene flow.
Right,
Yeah.
exactly.
Seems like a really,
I have 3 samples of Rubercyrus that still show over 1 million year divergence, so I don't know if it's just kind of a sampling artifact here. I definitely think in Hyoscyrus that there's going to be cryptic species, especially if you look at the distribution of this species in 2 totally different areas of Sulawesi. I just don't have the sample size to identify that, but I bet there's definitely more than 2 in Hyoscyrus and probably more than 1 in Rubercyrus. But maybe not. Maybe it's just a cosmopolitan, lower elevation genus.
there are not very many examples of that on Sulawesi.
Right.
If you have the sampling to screen Rubercyrus across all of Sulawesi, I mean, I'd bet my left hand you'd find a fair amount of deep genetic structure
Yeah.
across
It's
that.
still interesting that those 2 samples of my Murinus come from different parts of the island, and they are
Yeah.
less divergent
Yeah.
than the 3 samples of Weberi that you have,
Right.
which has a very restricted range.
Yes. And I had to actually.
Is not from any species that you sampled, is that
Not
right?
right, yeah, it's very different. So I don't know, perhaps it came from one of these other ones, or perhaps it's some ancient extinct animal, which there's,
Mitochondrial lineage at least.
right, or lineage, right, exactly. So yeah, it's kind of hard to untangle that with this sampling at least.
Are there any fossil lineages, extinct lineages that are
Well,
known?
are there any even subfossils on Sulawesi? I'm not sure.
There are
Fossil
some fossils from Sulawesi, but I don't know if they're fossil squirrels.
Yeah, I'd have to look into that. I haven't like tripped over that in the literature. So if anything, they're not abundant. But it would be interesting to look into for sure.
Are there must be plenty of squirrels in the Philippines. How do
Yes.
these things relate to this system?
Yeah, that's a good question. So I can, on this tree, so the Philippines has a lot of Sundasiris actually. And the Sundasiris genus, this is one I changed the name. It was actually originally in the genus Dromomes, which is a high elevation Southeast Asia genus. But in the Philippines, they have a bunch of diversity within Sundasiris that seems to have gone there multiple different times through Palawan. It's kind of stepped over because Palawan was connected to Borneo, and then somehow multiple times moved up into the rest of the Philippine islands.
Are they in Mindanao also?
I'm not sure if the same species is, but there are squirrels in Mindanao, yeah. Yeah, it's complicated everywhere, just like the Draco.
It's just, it would surprise me if if Mindanao squirrels were entered through Palawan and not through
Oh,
the Sulu
yeah, I see.
archipelago.
I'm not sure, that's part of that bigger paper I haven't finished yet.
Other questions? Missy, thank you very much.
Yeah, thank you guys.